Hello, everyone. Welcome to an ATP. My name is Aspa Khan, and
I'm Alexia. Hello.
today we're discussing the introduction in history to developmental biology. So before we begin, what do you think developmental biology is about, Alexia?
So, I think developmental biology is about how human beings came to be. So, it includes like the whole past and present of like our history in a more scientific perspective since it's bio.
All right. So developmental biology is the study of how a single fertilized egg can develop into these cell communities, and these cell communities become organized structure. And to understand development, we need to understand genes. This can be seen between the difference between embryology and developmental biology. So Alexia, before reading all of this, what was your opinion on what was the difference between embryology and developmental biology?
So, I thought embryology was only focusing on maybe like the embryo and only focusing on like that stage in like human life. Meanwhile, development would talk about like how you've developed and everything like that. So yeah.
Yeah. So embryology is mostly focused on morphology and the development of form in the different cells that arise from that, such as nerve cells and lung cells, while development is studying the cell biology and the genetic processes that create an embryo. So, why do we study development? We study development to understand more about humans, such as the fetus and complications that can happen during pregnancies and how to avoid that. Also, the same processes that occur in development also occur in cancer. So by understanding more about development, we can understand cancer and then come up with treatments. And regeneration is the study of stem cells in order to help create new limbs, like the newt and salamanders. Stem cells are cells that don't have any characteristics, like we have skin cells and nerve cells. Stem cells have no characteristics in whatsoever, and they can differentiate to anything. So, Alexia, what do you think is the most interesting to study out of?
Okay. So probably regeneration, since a lot of sci-fi comes from regeneration, and a lot of like what's seen on like TVs or movies, like superhero movies, especially like emphasize on regeneration and how there's a whole lot of research going into that could possibly benefit like maybe humans who are like disabled or who amputees who don't have like missing limbs and everything like that. So I would probably say that. But the fetus one is actually pretty interesting too, since it helps with like complications.
Definitely. So, Alexia, what is evolution?
Okay, so evolution is descent with modification. Descent with modification.
And how is it related to development?
Hmm?
How is it related to development, evolution?
It's related to development um because it sort of gives us like a foundation to build upon and like to research. And from our past, we can notice like how different we are from now and how we've developed into the human beings we are today.
Cool.
Big brain.
Yeah. So we use evolution for comparative analysis, as you said. So with descendant modifications, we have the same mechanisms in multiple animals, but evolution also creates diverse body plans. So you have different model organisms. So you have the fruit fly and the nematodes. And so with them, you can learn multiple different processes that can also be seen in us humans, because you can't do research about development on humans because that's pretty unethical. And so when you think about evolution, you often think about the common ancestor, and you think about the tree, right? Archaea, Prokarya, and and eukarya. And so when you think about bacteria, that's a single cellular organism, right? And it's going to stay single cellular, while might build up to multicellular organisms. So when you're thinking about bacteria development, those are going to be different life stages. And you're only measuring the cell density within a culture, and it's not creating tissues like eukaryotes. So in order to understand more about developmental biology, we need to study its history, so discover the origins of how the subject was actually created. So, Alexia, when was developmental biology first studied?
In the 1900s, the 1800s,
It was
I would say.
the 5th century BC, during the Greek era, and one of the most prominent people to actually study developmental biology was Aristotle, surprisingly. And he was able to help us determine one of the major points, which was preformation versus epigenesis. So preformation is the belief that that all of the tissues and all of the organs are already created when the sperm fertilizes the egg. However, epigenesis is when is the belief that the tissues are formed throughout development, and that's what Aristotle believed. However, when the religious era came, lots of people went back to the preformation belief because they couldn't wrap their minds around epigenesis. And what do you think? What do you think helped support the belief of epigenesis after people stopped believing in it?
I'd say the cell theory. I'd say the cell theory, especially since the worksheet has it right under the epigenesis. So probably the cell theory.
And what are
Yeah.
the main 3 tenets of the cell theory made by Shieldon and Sean in the 19th?
Okay. So I know number 2 is like the basic unit of life. Oh my god. How could I forget this? The all living organisms are made up of cells, and then all cells came from like pre-existing cells or something like that. Okay. Oh Jesus.
This was really helpful for understanding more about developmental biology, which can be seen because the division of new eggs create new cells. So the last one was especially important for that one. And so with mitotic divisions, we have new somatic cells from the zygote. And it's important that we have the emphasis on the somatic cell because it was Weismann in the 19th century who said that the characteristics that the embryo has is determined from the germ cells. So any mutations created in the somatic cells are not passed down. And that's really important to know, especially in the genes that help with development. So when we're looking at Weismann's germ cells, uh, a quote comes up from the novelist Samuel Butler. A hen is only an egg's way of making another egg. So in terms of development, the body is merely the carrier of the germ cells. So what next time you're thinking what came first, the egg or the hen, it's the egg. So over here we see the cell theory, and the cell theory helps us with development because we see the single cell creates multicellular organisms, and those multicellular communities create the embryo through organization. So there are 2 types of development. The first one is mosaic, and that was discovered by Weismann in 1880. And so mosaic development states that there's determinants within the nucleus, and by the cell division, the different proportions of determinants are within certain nucleus, and that determines behavior and cell fate. This was also supported by Roux with the frog. He used a hot needle and he poked into the half of the larva, and that created a half larva. But it was determined later that that's not actually nuclear determinants, but cytoplasmic that determines the cell fate. And we can see the Roux experiment here. He takes the hot needle in one half, and it creates half a larva. However, Regulation is another type of development, and this was discovered by the Jersh, who did the same exact experiment as above, but with the sea urchin. And he found regulation, which was the ability of the organism to restore normal development early on. And we can see that here, where when half of it is taken away, the cell is able to use its own information and through regulation and create the entire larva just smaller. Do you have any questions about this, Alexia? Anything?
Okay, my question now is like when it says like the ability of an organism to restore normal development early on. So we're like the larvae and the sea urchin like at different points like in life. Like can the larvae restore like normal development early on, or is that just for like the sea urchins?
Um, no, it's dependent on the organism. So we talked about evolution, descent with modification.
Mm-hmm.
So like with those modifications, the sea urchin is able to do this, while the frog is more dependent on mosaic development rather than regulation.
Okay, so these aren't like
That's why we
theories.
study,
These are like legitimate types of development that you see in living organisms.
yes,
Okay.
that's why we study multiple ones. So, an important thing to help us understand more about development is cell communication, and um, that is the ability of induction. So one cell can direct the development of neighboring cells. This was seen in 1924 with Spearman and Mangold. They found a group of organizing cells and that were responsible for controlling the organization of the larva. As you can see here, the king is the organizer and the cell that's smoking a cigarette is a rebel and he won't conform to the king's expectations. So what's really important to understand development is genetics. And it took a really long time for genetics and embryology to come together to create developmental biology that we know today. And developmental biology is understanding how genes are expressed to create a functioning embryo. And genes are able to control what proteins are expressed. And so when they control what proteins are expressed, they control behavior because proteins are really important for behavior, cell behavior. And so to study developmental biology, we use model organisms, as I said before. And the model organisms that they use are easy to obtain and to do research on. And the type of research they do is transgenic research, which is manipulating genes. And that's really easy to do on the fruit flies. And the reason that we can use model organisms is because of homology. So because we had a common ancestor, it is believed that over time, even though we have evolved to different organisms, we still have the same genes because of our common ancestor. So the way that we study the specific genes in developmental biology is through mutations. So mutations identified in development can either be spontaneous or induced. And so we see the mutation first and then we see the gene behind it. And that's called forward genetics. And mutations in invertebrates need to be conditional, so they need dependent on temperature and environmental factors. And new techniques in the modern era include sequencing because we have a lot of sequenced DNA sequences of animals, and we can see any errors that occur, and then we can see the mutations that arise. Or we could do reverse genetics, in which we knock out a certain gene, silence it, or with the CRISPR-Cas9, we can add or take out a gene and then figure out what mutations arise. So now that we've learned more about developmental biology, Alexia is going to give us a summary about it.
Okay. So developmental biology. So developmental biology is focuses on like the genes in a single fertilized egg, right?
Yeah, genes, also cell signaling as well.
All right, genes, cell signaling, and a single fertilized egg, and then its stages.
Yeah.
This is different from embryology, right?
Yeah.
This is different from embryology because embryology would focus on morphology and sulfate, I think you said, right?
Mhm.
Yeah, right. So it's different from that perspective. Um, let's look at my notes. All right. So why is it important? It's important because it can help us uh, do research such as fetus, right? Such as like fetal complications and like the complications during that development, cancer treatments, and also like the regeneration, um, and how it can focus on like stem cells and like just everything involving that. What else did we talk about? We talked about how there are model organisms that we focus on, and how these model organisms can be are we use model organisms that are like easy to use and easy to sort of like follow through so that we can study them further. We talked about how there are two types of development, which is regulation and then mosaic.
Yeah.
And mosaic would be, I don't remember that. Regulation would be how it's able to, it's not, is it able to regenerate?
It's able to restore development early on. So when you poke the hot needle, it can use
Yeah.
the information it has in order to restore what it lost.
All
Whereas
right.
with the determinants, you lose it, you don't
Mm-hmm.
have it anymore, the determinants are lost, and so
Okay.
it's not able to restore development.
Okay. So regulation would be restore, mosaic would be you lose it. You lose the determinants and everything. Okay. Restore, lose determinants. We talked about evolution and how descent with modification helps us understand our common ancestors and how that's different from our biology that's developed today. Um, what else did we talk about? Mutations,
Yes,
right? And
that's
how
how you study.
genes came, it took a long time for us to like connect genes with developmental biology. But once we figured that whole route out, it helped us into this whole new avenue of research that helped us with developmental biology, right?
Mhm. Yes.
I wrote notes on that, but like,
Yeah.
yeah, that's all I
But like
have.
final like one thought like about this section that you would say.
One thought about this section. Hmm. I would say, hmm. One thought. Oh, right. At the end of the worksheet, it mentioned like what is like absolutely essential for um developmental biology. Like what is very important to help us to understand developmental biology. And I didn't know if that was like genes or if that was mutations.
They go
Because I
hand.
think
So genes is what is important to understand, and you do that through mutations.
okay.
Yeah.
So
Yeah, the
mutations would
genes
be like
function
the thing
without
before
mutations.
genes.
That's why, actually, fun fact, genes are named after their mutations. So it's gets
Okay.
really confusing when you actually study development in Drosophila and you're like, it's called dorsal, but it doesn't create the dorsal. It creates the ventral. Because
T.
without it, you have a dorsal. Yeah. Genes. All right. Thank you for watching. Hope you see the next one.